Welcome to Medicine Handy Point. The topic is MRI Brain 4 Simple Rules which will help you understand almost 80% of the MRI. The target of today's video is to discuss different sequences of MRI Brain simple rules. That are 4 rules that will help you in identification of different sequences. 5 main MRI sequences used are T1, T2, FLAIR, DWI, and GRE. There are many others, but these 5 are more important. Rule number 1 is T1 and T2 are opposite to each other. Let's do some examples. In T1, white matter is white and CSF is black, while in T2, white matter is black and CSF is white. So just memorize rule 1. You will understand almost all of the MRI sequences. In T1, another example, hemorrhage is white and ischemia is black, while in T2, hemorrhage is black and ischemia is white. Just memorize this rule 1 and you will cover all of the MRI sequences. Now let's apply rule 1. Image A is T1 and you can see that the white matter is white and CSF is black. Image B is T2 and you can see that white matter is black and CSF is white. So rule number 1 applies here. Now this is the MRI sequences of the hemorrhagic stroke patient. Image A is T1 and you can see that the hemorrhage is white here, while image B is T2 and you can see that the same hemorrhage looks black on T2. So rule number 1 applies here. They are opposite to each other. Now this is the MRI sequences of patient with ischemic stroke. A is T1 and you can see that there is hypodense black area of ischemia on T1. Image B is T2 and you can see that the same ischemia look hyperintense white on T2. So rule number 1 applies here. Now let's discuss rule number 2. T2 and FLAIR are the same, but in FLAIR CSF is made black and there is a reason why CSF is made black. We will discuss it later on. So let's see the pictorial presentation of the rule number 2. So let's see rule number 2. In T2 the CSF is black and while in FLAIR the CSF is made black. Both of these are T2, but in FLAIR the CSF is made black so that any lesion there near the ventricle can be delineated. Now this is MRI sequences of patient with ischemic stroke. A is T2. You can see that there is ischemia near the ventricles. You cannot delineate ventricle in ischemia, while in B image is FLAIR and you can easily delineate ventricles from the ischemia. So this was the reason for FLAIR to properly delineate the ventricles from the surrounding lesions. Now rule number 3, DWI is same as FLAIR, but DWI is a kind of microscopic version and detects early changes, especially ischemic changes. Now you can see rule number 3. In the FLAIR you can see the ventricles are black. DWI also have ventricle black. They are almost the same, but DWI detect only ischemia. Now this is the example: ischemic stroke patient. Now within few hours you can see that in DWI the ischemia can be seen as hyperintense area, while in FLAIR T1 and T2 the ischemia is almost developing but not very clear, while in DWI it is already developed. Now the rule number 4, which is the last one, is GRE is T2 with ability to detect early hemorrhage, just like DWI detect early ischemia. So GRE, which is the gradient echo sequence of MRI, is actually developed to detect early hemorrhage. Now let's see the pictorial presentation. You can see CT scan or 2 different CT scan are having hemorrhages, and in the end you can say the GRE sequences are showing those hemorrhage as black spots. You can see it. So GRE is just like DWI, and GRE is used to detect early hemorrhages. Uh, I hope it will help you. Please like the channel and please subscribe as well. Thank you.